And so what happens is in the cold, then the reaction rate goes down, but at a slower rate as the temperature gets colder, because the enzymes themselves and all the molecules in the cell are moving slower. So if you have an enzyme in its substrate and they're bouncing around, all right, they have to randomly come into contact with one another. If it's cold, that bouncing around, that moving around is a lot slower, so they don't come into contact with each other as much, and therefore the reaction rate goes down. All right, so that's the difference there. And this just shows you how in different organisms there are bacteria that live in hot springs and very hot environments, and their optimal temperature is much, much higher, which that's why they're able to live in that particular temperature and why we can't. All right, and so they have different enzymes with different optimal temperatures. And then the bottom here shows two examples in humans, the pepsin and the trypsin, two different organs in the stomach and in the intestine that require a different optimal pH. And so here in the stomach, it's acidic pH, and in the small intestine, it is basic. And so that's why our stomach releases acid. The acid creates the acidic environment so that our enzymes in our stomach can actually digest your food. All right, so it's the enzymes that are digesting your food. The acid is kind of making them have the right optimal pH for digestion. There are other things that play a role as well. There are cofactors. Cofactors are non-protein enzyme helpers, and you guys don't have the examples here, but you might want to jot down just to give an example. I think I wrote zinc, I think, on there. Did I write zinc?
Mm-hmm.
Okay. All right. Iron, magnesium are examples. Collectively, metal ions are examples of these. And then coenzymes are organic cofactors. Organic means it's a molecule that contains carbon. A lot of our vitamins are like this, water-soluble vitamins.
Does that involve, like, when you say a lot of vitamins, does that involve all vitamins?
No,
or certain
no. So, I gave some specific examples, but it's not all of them. No, all right. So, water-soluble vitamins. Next chapter we're going to look. These will come back. All right, NAD plus and FAD. I put it here, derived from niacin and comes from riboflavin. That's what you'll see. Like if you look at what vitamins are in, look at the nutrient labels on foods and things like that, you might see the words niacin and riboflavin. What they do is our body uses those to make these cofactors. And so both these guys here, the metal ions and the coenzymes, both of them help an enzyme to work. So therefore, some enzymes, in order to work properly, have to have certain metal ions that bind to it, and it doesn't, and that puts it slightly in the right shape. So the enzyme won't work properly without those things. So having those things in our diet is very important to make our enzymes work properly. And the same thing with these coenzymes. So some vitamins help the enzyme to work properly. And so some enzymes need some help there. All right, so they have to be at the right temperature, the right pH, and some of them need some other molecules to help with that. And then the last thing with enzymes that we're going to talk about today is that some enzymes can be inhibited, so they can stop they can be stopped, and so therefore stop the reaction that they catalyze. There's two kinds. Competitive inhibitors bind to the active site of the enzyme and compete with the substrate. And then there's non-competitive inhibitors that bind to another part of an enzyme, and that causes the enzyme to change shape, and therefore the active site is less effective or may not work at all. And so this is what this picture shows here. So notice here in the purple here, this is your enzyme, it's labeled enzyme, and this is your active site where the substrate can bind. So all of this is what we've talked about here. So the substrate is going to be converted to product once it binds to the enzyme. One thing I want to point out that's new is this little guy here. It's another site that's different than the active site. And I'm going to draw a line to that. And this is called, this other site that's on a separate spot from the active site, that's called an allosteric site. draw a line to that. That's going to become important down here. Not all enzymes have that, but some enzymes do. All right, so this is the normal binding. So the next two pictures show you what happens with the two types of inhibition. So a competitive inhibitor, what a competitive inhibitor is, it's called that because it has a similar, not exact, but a similar shape to the substrate. So remember that enzymes are very specific to what substrate it's going to bind to and then therefore convert to product. And so in this case here, this competitive inhibitor has a slightly similar shape and therefore can bind to part of the active site. And so what happens is this competitive inhibitor and you have the substrate, if both of them are bouncing around in the cell and then you have the enzyme, if the inhibitor bounces into the active site first and gets to the active site first, then when the substrate comes over here, that's what this little arrow here is showing, that the substrate cannot bind there. And so therefore that reaction is not going to occur. And so they're competing. It's called this because they're competing for the same site. All right. And so then if the competitor gets there first, the substrate isn't converted to product and it slows the reaction down. This is reversible though. Often the competitive inhibitor can come off and then the enzyme can work again, and if it bounces, it comes into contact with the substrate, it can bind there. All right. And so that is reversible. And then the non-competitive inhibitor, doesn't compete for that active site. So when the non-competitive inhibitor binds, it binds to that allosteric site, and so it's away from the active site, but it leaves the active site open. But what has it done to the active site when this binds to it? Changed the shape, and so now the substrate still can't bind to the enzyme. So therefore, this is still an inhibitor because it's inhibiting the enzyme from working properly, but it's non-competitive because it's not competing for the same active site, but it can still change the shape. So it changes the shape and doesn't allow the reaction to occur. And this is also a reversible process, so this can come off, all right? And so sometimes, though, there are situations that it can be permanent. And that's when the inhibitor, if the inhibitor covalently binds to the enzyme, it'll stay put and it'll stay there and inhibit it forever. So there are situations that it can be permanent. And so just to give you an example, like certain poisons work that way. And so we actually use this in a lot of insecticides when we spray for insects and so on. Those chemicals can be, some of the ones that we use are competitive inhibitors for certain reactions. So they bind to and permanently attach to enzymes in the bug, in the insect, in a pathway that if it doesn't happen, that reaction, they die, all right? And so therefore it kills the bugs, all right? And so we use this to help us a little bit with insecticides. For us, some poisons and things like that can be deadly because of the active inhibitors and render certain enzymes useless because they cannot do their job. All right, and so then whatever reaction that was supposed to catalyze is not happening and can have detrimental side effects. All right, and also there can be inhibitors that are actually a good thing. We actually control some of our reactions in our cells by purposely inhibiting enzymes. So sometimes, not all of our chemical reactions we want to occur all the time. And so we can control when and how much a reaction can occur by controlling the enzyme and making the enzyme work or not work. And so we can control chemical reactions in our cell that way. All right, we'll talk more about that tomorrow. This is where I want to stop for today because I want to look at and have you look at the sheet that you picked up today. I'm going to do a little activity that's going to illustrate a few things. And so I'd like you to look at and read the first page, please. All right, let me talk a little bit about this and make sure everybody understands a few things. So you're going to do this activity that's going to simulate and illustrate a couple of points that I want you to get about enzymes. And so there's two different trials that you're going to do. So you're going to be an enzyme, and the substrate are the toothpicks. And so you're going to go through the first trial as it says, and your hand is the enzyme. You only use one hand. You're going to work with a partner, and you have to pick which person is going to be the enzyme, because we need to have the same person be it for the whole time, or else we're including another variable in our data, because some of you may be better enzymes than others. All right, so therefore, in yours, the reaction is breaking the toothpick in half. And so you're going to have 40 toothpicks. Just leave the toothpicks alone for a second, okay? You're a bowl, and the bowl is your cell, and you have the 40 toothpicks in there. You are the enzyme. You cannot look. The enzyme cannot look, because enzymes can't find their substrate. They're moving around randomly in the cells. And so you don't look, and you pick one up one at a time, because an enzyme can only react or bind with one substrate at a time. And then with one hand, you can break the toothpick like that, all right? And then it goes back in there, all right? So the products go in the cell, all right? So it goes back in there, and then you pick out another one, all right? All this time you're not looking. And so you're going to record. It says for the first 10 seconds. And so if you look at the back of the first sheet, there's a data table. And the data table has the time in seconds, and it has trial 1 and trial 2. So we're talking about trial 1 right now. At time 0, everybody has how many broken toothpicks?
0.
0. So everybody's putting 0 there. So then you're going to use your cell phones for timers. So the person who's at the enzyme is the timer. So you're going to put how many, at the end, you're going to literally look in your bowl and count how many toothpicks were broken. So let's say you broke 4 in the first 10 seconds. So I looked, all right, for 10 seconds. And so then, so I broke 4. I put everything back into my bowl. So all my broken toothpicks, because now my bowl as it sits is as if it's at the 10-second mark, right? And so then for 30 seconds, rather than getting 40 new toothpicks and going from 0 to 30, you now have your bowl in a situation where it's at 10 seconds. Now you can start your timer for an additional 20 and continue to break, all right, until the 20 seconds are done. Now in your bowl, how many broken would be representative of after 30 seconds? Does that make sense? All right, and so then when you count this, you're counting total broken after 30 seconds. So it's cumulative, so that your 4 you put back in, and those 4 are still part of your data for now for your 30 seconds. Does it make sense? So your number should go up, okay? And so then for trial number 2, to illustrate a few things here, as you're working on trial number 1, I'm going to come around and assign different groups a different scenario. So some groups I'm going to assign and say for trial number 2, you are going to increase the substrate concentration. So what we're going to do by increasing the substrate concentration, we're going to increase it by 2. So for trial 2, you're going to start out with 82 ticks, all right? For the enzyme concentration, how could we simulate that?
More people.
2 people, or
Both hands.
same person, 2 hands. I'll let you pick what you want to do, all right? So you could do either 2 people or 2 hands. And then what we're going to do for the competitive inhibitor. The competitive inhibitor is going to bind momentarily to the enzyme. And so I wanted to have something where you couldn't tell by feeling what the competitive inhibitor was. So what we're going to do is, and some of you that I signed this, I have 2 groups that I already have made up, but some of you will have to make up your own toothpicks if I assign you this. What I had them do is we're going to take 10 additional toothpicks, and I had them just mark it with a black Sharpie marker in some way to distinguish those toothpicks from the other toothpicks. And so then what you're going to have is you're going to have the still 40 toothpicks, and then we're going to, so that we can compare it to the first trial, and then we're going to add 10 with the black toothpick mark on it. So then what's gonna happen here is you still can't look if this is what you're assigned to, and so you pull it out, your partner, you can't look, your partner needs to look and say, you can't break that one. If it's a competitive inhibitor, enzymes don't do anything to the competitive inhibitor. What the competitive inhibitor does is just stop the enzyme from working for a little bit, all right? So therefore, when you pull it up, your partner says nope, and you have to put it back down and pull out another one until you get one that's not a competitive inhibitor, and then you can break it, okay? All right, and then you do your data. Then after that, if we look as we go on in our sheet, after you get your data in your packet, the next is a graph, and it's going to be a line graph, and you can put both lines on the same graph there. And then if you look at the next page, you're going to use your graph and use your data points, and the next page shows you how to figure out the rate of reaction, which is really the slope between your data points, all right? And so you're going to look at your graph and figure out the slope between 0 and 10, and between 10 and 30, and so on and so forth. And then there are questions, okay? All right, so everybody understand what we're doing? Okay, so then let's